Over the past 4 years, my lab has been developing high-throughput omics approaches to investigate the small molecule metabolite complement to the genome in blood and blood components. When the pandemic exploded, being sick from COVID myself at the beginning of March, we decided that our methods could be immediately applicable to investigate the impact of SARS-CoV-2 infection on COVID patients.
I am part of the multidisciplinary team of investigators that focus on performing multiple different assessments of the COVID patient samples to understand immune responses. Particularly, my team focus on understanding the immune cellular changes in the context of COVID infection. By understanding what immune cell types change and how they respond to infection in the context of COVID, we can begin to formulate hypotheses as to what immune cell types we think are important in establishing a protective response.
I have the great fortune to have been the final link in a chain of coordination and collaboration that extended across campus to bring forward an antibody test for SARS-CoV-2 that was completely supply chain independent and based on the expertise and what we have on campus here. And together we came up and in the end in my lab brought to a clinical validation, a CAP-CLIA level assay that we could use to look for antibodies to SARS-CoV-2 independent of what else was happening in Colorado, what demands there were nationally or globally for those reagents, because we could make them here on campus and use our expertise.
We developed a dried blood spot-based assay so that people could self-collect their own blood samples for looking at antibodies, so that it requires not having a trained phlebotomist. And we validated that assay using this multiplex serology assay, and that assay is going to be really important because we can measure 2 different antibodies to SARS-CoV-2 in the plasma, and that allows us to distinguish between vaccine responses versus a natural infection in a community surveillance when the vaccine ultimately gets here.
One hallmark of the COVID pandemic here in the U.S. has been just the extreme variation in how it's played out across the country. And we've studied characteristics of communities that have made them more resilient, helping to slow the spread of the disease and also reducing the adverse health and economic outcomes, particularly for vulnerable and historically marginalized populations. And the key finding that we found here is that it's the strength of the networks that exist between medical care, public health, and social service organizations that really has been effective in helping certain communities be more resilient to the pandemic.
We are conducting a number of clinical trials looking at therapeutics for treating patients with COVID. And our hope is really to figure out effective therapies now. These are move-in ready treatments. So we're truly looking to find things that will prevent people from progressing to severe disease, saving lives, as well as those who do recover, helping with their recovery, make it better, faster, reduce the burden of symptoms, get them back to work and life as soon as possible.
My role in the COVID research effort has been to orchestrate clinical trials for COVID treatment and prevention here on the Anschutz Medical Campus. We have conducted studies of COVID treatment both in the inpatient and outpatient setting, including drugs such as remdesivir, as well as the Regeneron monoclonal antibodies.
This study was a great partnership with the Arapahoe County Office of Emergency Management and who reached out to the campus to have help in understanding the seroprevalence of SARS-CoV-2 within the community and the first responders.
At the start of the COVID-19 pandemic, my laboratory quickly mobilized to establish a SARS-CoV-2 laboratory at BSL-3 to support COVID-19 research efforts here on the Anschutz Medical Campus. Our efforts during this COVID-19 pandemic have contributed to a number of different research manuscripts, as well as 25 or more grant applications for funding, 1 patent application, and the development and implementation of diagnostic assays that are currently being used here in clinical laboratories at the Anschutz Medical Campus.
Over the past 3 months, we had a chance to publish now 5 papers on the impact of COVID-19 on the serum metabolome and proteome, but also on the red blood cell proteome and metabolome. A paper on damage to red blood cells has been highlighted by the New York Times. Specifically in this very paper, we did find that damage to red blood cells can result in changes in red blood cell membrane components. And since red blood cells are devoid of protein synthesis capacity, they cannot replace damaged components while they circulate in the bloodstream.
We have lots of collaborators here on campus and across the country. On campus, we've been working closely with colleagues in the Department of Family Medicine here in the medical school, also with colleagues over on the Boulder campus in the Natural Hazard Center. And we've also got colleagues across the country, in particular for the Preparedness Index work at colleagues at UCLA School of Public Health and at New York University.
The collaborative effort that has been in place, the mobilization of our resources has been tremendous, and without this 360-degree support for this research, we would not have been able to make the progress that we've made so far.
We could not do these projects without the spirit of volunteerism and altruism that comes from the patients and the families that participate in these studies.
You know, to see a pandemic and to be able to step forward and do something is really gratifying. And I have to say, this collaboration and the strength of it is going to probably forever be a highlight of my career.